the first part, it is on a physical problem on which some experiment has been done by us, and we have some understanding of what is going on. There isn't much of a mathematical description there. The second part would be on mathematical treatment of a class of sort of problems in biology. And for that, in both these problems, the common thread is that we look at the first passage question. And there is no deeper sort of connection between the two parts, only that there is an apparent sort of thing that we see which is similar, which is that the fluctuations, the relative fluctuations, they show some non-monotonic behavior. This is why I thought that maybe I can talk of the two things together in a single talk. Okay. And the second part deals with biological timekeeping. So I will explain all those as I go along. So, we would start with this familiar picture of a textbookish example of a random walk in which you have a drunk person moving on the street in some random fashion. And then you say that it starts from a point, a blue point, and if it hits the lamppost, which is marked by a red point for the first time, then we say that first passage event has happened. The time taken for this walk, starting from the blue point to the red point, is called the first passage time. Okay, so so given this classic, every textbook you open in, they will start talking about a drunk person. It's a little misleading because the drunk person is never dead drunk. And the person actually has some sense of the home or the shelter to which he has to return. So in some sense, he is directed or, self-propelled in a way. And then we can have variants that, the target need not be a point. It can be that when this line is crossed for the first time, you can make it that also. And it may also be that there are cobbles and rocks or stones on the path of the walker. Okay, so this is some kind of version, an abstract version of the problem, but you don't deal with drunk person or stones and all that in the lab. What we have dealt with is something like this. So the stones are now replaced by some purple discs that you see. Those are paper discs that are put on the surface of water. Okay. And then instead of the drunk person, what we have is something called a Camphor Boat, which is a self-propelled particle. Why it is self-propelled, you will get to see in the next slide. And the target here is the whole circumference of the Petri dish. So anytime for the first time the Camphor Boat reaches from the center to the periphery of the dish, the process ends. That is the first passage event, and you would record the time and start once more and then record the time again, and thus you will get statistics of times. Okay, so this work was done by my PhD student Animesh, and in the lab of my colleague Puneet Parmananda, and there was a visitor Manuel Cruz in his lab who assisted Animesh in his works, and the paper has appeared in Soft Matter 2020. So the paper disc is to be coated with camphor, but camphor does not dissolve in water, it dissolves in ethanol. So this disc is soaked in that, then taken out and dried, and then you have a disc which is basically loaded with camphor. And then you release it with a syringe at the center of the Petri dish, and then with the camera, the trajectories are recorded and all that. And this is the top view, the circular disc you can see at the center of which the Camphor Boat is released. And then this is the side view on the left. And what you can see here is that as the Camphor Boat lands on the surface of the water, the camphor particles are released from it. They move on the surface, they spread out. They also evaporate, some of them sink down. Okay, but now this the moment these camphor they start getting into the water, the camphor lowers the surface tension of water. So you would agree that at the center of the disc, the surface tension will be lowered more, while away from the center of the disc, where the concentration of the camphor is lesser, the surface tension will still be high. So there would be a surface tension gradient that will develop. And this leads to the classic Marangoni force, which then propels the disc. But of course, the disc is circular, and if everything was perfectly symmetric, that is the release of the camphor in all directions were symmetric, or the disc was perfectly ideally symmetric, then there would be no force, because all forces from all sides will cancel. But real life is never ideal. I mean, you will have a disc with some kind of random asymmetry somewhere, and the camphor will release in directions in unequal ways, and hence there will be a resultant gradient which will propel the particle, and that's what makes the camphor boat a self-propelled particle. Okay, so here is the video. As you can see, the particle is moving in circles, and it is moving kind of in a noisy circular fashion. What do I mean by noisy? Its center and radius, you see, are all changing as a function of time. Finally, after some time, it lands on the circumference. So just now, whatever you saw in the video, this is the trail of that. The green curve starting from the center ends at the point, the red point. But in the meantime, it has gone like this. And now you will appreciate my statement that it is a noisy circle. Its center as well as its radius are fluctuating with time. And this is the x coordinate of this motion. You can also plot the y coordinate. They are oscillatory, but the amplitudes as well as the center about which it is moving, those are all changing with time, as you can clearly see. And this kind of noisy circular motion, I mean, this is a noisy circular motion, but those who are familiar with active matter may know that in microtubules on motors, that kind of assay, people have seen circular motions. With motile bacteria in 2D fluids, circular motions have been seen. So this is a chemical sort of analog of such biological systems, if you wish. So just a brief slide. Question is that what all theoretically is known about this? This is the one paper that has attempted to do some theory, and I will kind of tell you that where it goes to, not very far. So the point is that if you want to write the dynamical equation for the center of the disk, the position of the disk being rho, so mass per unit area into acceleration is equal to the drag force, that's the first term, xi d rho dt, and then there is this so-called Marangoni force. So how does that come? On the top, you may see that I have shown a force expression, which is surface tension integrated over the circumference of the disk. Now, since the disk is very small, you will Taylor expand this, and that's how the gradient will come. And the surface tension is proportional to the concentration. That's a basic sort of assumption. Within a certain concentration range one can make. And if that is so, then the gradient of the surface tension becomes the gradient of concentration. So that's how the position of the disk is coupled to the concentration of camphor. On the other hand, the camphor concentration evolves in the following fashion. There is a diffusive term on the right hand side. Then there is a decay term of concentration. Why decay? Because some of the camphor evaporates or sinks. And there is a source also, which is coming due and from this Dirac delta function. So why a source? Because from the center of the disk, which is at the position rho, the camphor is being released into the water. So you see the concentration equation is coupled in this non-trivial way to the position of the disk. The position of the disk is coupled to the concentration. There are boundary conditions on this concentration, and all these have to be solved. So of course there is no exact solution. These people attempted to do it sort of approximately and got a nonlinear equation for the position rho, and a stability analysis of that showed that yes, stable circles are possible when the viscous drag is not so high. If the viscous drag is high, the stable point is just at the center where you release the particle, it will remain there. But of course, with reasonable, situations that we have in experiments, circles are possible. So this is all that is known theoretically so far. Nobody has done any further theory. And the point is that this also is incomplete. You see, we have noisy circles. So these equations one has to put in noise. Now, where do you put the noise? Because the noise can be at various places. The noise can be at the place where it is even being, the source term is being added. Or, where do you put it in some, on the first equation and all that. So that remains an open question, and that has not been addressed. But this is just, because some people may be interested that is there some mathematical analytical way something has been thought. But the rest of this part of the talk will have no further mathematics. So you see, now our interest has been to look at the motion of the self-propelled particle, but in the presence of these disks, which are these purple disks. So just now I showed you the video when there were no disks. You remember the noisy circles? That is when the packing fraction of the paper disks is 0. Now let me go to a very crowded situation when the packing fraction of the disks is 0.44. Okay, so that is somewhat dense environment, and that is where this video will show you that as the self-propelled particle is trying to move, it is rattling in cages, and then it sort of you can see that I am forwarding the video. After some time it sort of comes to the periphery, and finally it reaches the periphery for the first time. So now the trail on the side, you see, no longer has those noisy circles. It has become random motion. What kind of random motion? We would be interested to know. And this is this x coordinate of this trail that has been plotted at the bottom, and this is some kind of random motion. So if you go and look at the mean square displacement, you will see that as expected it's more like an active Brownian motion. That is, up to some time it is having a ballistic behavior. No wonder, because it is self-propelled. So up to some distance it will go ballistic. That is why the power is 1. And beyond some time scale, the power is half, which is classic diffusive. And merely the mean square displacement being half delta t to the power half does not tell me it is diffusive until and unless I show you that the displacement distribution is also Gaussian, and that can be nicely collapsed by the scaling. So it is definitely diffusive at long times, and that is what is happening at very high densities. So now the question, we were not interested really in transport. Our question was that what happens to the first passage times and their statistics, their fluctuations, as we go and tune the density of these disks. So we go from the left to the right. Left is the lowest density, right is the highest density, and in between we studied 3 densities gradually increasing. That is 11%, 18%, 32% field packing fraction. Okay, so now for every packing fraction, these 5 ones, we did separate 5 experiments. All the water is thrown away, everything is redone 100 times so that we get statistics. Okay, so what is it that we may expect? So you see, anybody would expect that as the crowding would increase, the particle will find it more and more difficult to reach the boundary quickly. So as a result, it is expected time scales, all time scales, and in fact, the mean first passage time should also increase with packing fraction. And that is what we find. Very much expected, because packing fraction goes up, mean first passage time should go up. What is the surprise? But then this is what we did not expect. What we found was that the relative fluctuation, that is, here what we have plotted is called the Fano factor, which is variance of the time divided by mean. Okay, this is one of the markers. One can look at CV square, which is variance divided by mean square. Different measures of relative fluctuation are there. This is Fano factor. And this actually has a high value at small values of the density. It has a high value at high value of the density. And at intermediate densities, it goes down. So that means that at some intermediate density, which we can call as optimal density, the fluctuations will be the lowest. Now that is a bit strange, because why it is happening, and then one would seek answers in things. But before we went ahead to ask why it is happening, we wanted to be sure about it through another characterization, another quantitative measure, and this is what is called the distribution of uniformity index. Okay, so what is distribution of so what is this uniformity index? So anytime you have random some set of samples which are random, you can always try these, and this is a very nice quantity. So in our case, the samples are the sample times, the first passage times, right? T1, T2, T3, various one of them. So you pick up any pair of such random times. Let us call one of them as T1, another as T2, and construct this quantity T1 divided by T1 plus T2. Now, if the time T1 is much lesser than T2, then you are going to have omega close to 0. On the other hand, if T1 is much greater than T2, you are going to have omega close to 1. On the other hand, if T1 is very similar as T2, you are going to have it as half. So you see how nice this marker is. If you are having less fluctuation in the system, then your times will be close to each other. Then you will have a distribution of omega picked near omega equal to half. On the other hand, if you have large deviations away from each trajectory is very different, and you have lot of fluctuation in the times, in that case omega will have weightage near 0 and 1. So this is what we find. So for low densities, you see that there is a lot of weightage also near omega equal to 0 and 1. Of course, there is weightage near 0.5, but there are also non-trivial weightage near 0 and 1. Similarly, if you look at 0.44, the high density, you will see that rather than weightage at 0.5, if you move at, for example, 0.25 or 0.75, there is considerable weightage actually. On the other hand, if you look at some intermediate density, you see that it is a unimodal curve essentially. No, within sort of reasonable experimental fluctuations, nobody would disagree that the middle curve has a single peak around 0.5. So, this kind of thing that the fluctuations are higher for low density and high density is now clear through this measurement too. So now what is going on? So what one realizes is that if one looks at the trajectories themselves, one gets an insight very clearly about what is going on. So you see, you can produce these noisy circles. Now the circles sometimes can have big radius, and if they have big radius, then it will reach the boundary immediately. So it will be very short time. On the other hand, if by luck the radius of the circles remain small for a long time, they are not going to reach the boundary for a long time. So there is going to be long trajectories. So short and long trajectories will be there a lot. Now what you are doing is that you are putting paper disc on the water surface. So the paper discs are not going to allow the particle to have this kind of circles anymore. They are going to cut those down. Okay. So you are now going to have these paper discs which are coming in to create a noisy environment, but they actually cut down the initial source of noise. The initial source of noise was these noisy circles. Those circles are getting eliminated, and a new sort of noise is being introduced because the self-propelled particle has to go past all these obstacles, and that will take some effort, and that introduces a new source of noise. So the old source of noise is increasing, the new source of noise is the old one is decreasing and the new one is increasing. And this is what eventually happens, is that when you go to even higher densities, there the crowded environment can produce its own fluctuations, which are sometimes short trajectories or long trajectories. So the fluctuations actually go high. So you have high fluctuations at the two ends and low fluctuations in the middle, as a competition between two sources of noise, one due to the paper discs and one due to the inherent dynamics that was already there for the self-propelled particle, and that produces an optimality. Okay, so this is now the concluding remark, that this kind of confined geometry, crowded environment, active particle has been studied by other experimentalists, but per se the first passage question was not addressed, not known to us, at least before our work in this kind of setup. And then what is novel about our work is that we saw these non-monotonic fluctuations of the first passage time. Well, people have looked at non-monotonic behavior in the mean in various other contexts. I have given two examples. I mean, famous one being stochastic resetting, for example, in the stochastic process literature, or this bacterial system. People have looked at path lengths. They have found non-monotonic path lengths as a function of crowding, as well as mean time having non-monotonic behavior. But what we are telling is that mean time is monotonic. The fluctuation is itself non-monotonic. So that is the interesting thing that we find. So I will now go to the next part of the talk, but I can stop for a minute, and if there is any question for a minute, I can take.
So there is a equation written at the beginning to describe these paper boats, and is it possible to describe these three regimes in the equation itself?
Can you once more tell? I completely could not hear properly. Sorry about it. Little loudly, will you tell?
Just 1 second. Yeah, maybe someone else can ask a question.
No, just go ahead, PK. I just could not hear properly. Once more, I'm happy to answer.
No. Happy to answer. I realize what is the mistake. So can you hear me now?
Yes, it is better.
Yeah. Okay. So so there was an equation written for this.
That is for a single self-propelled particle.
Oh, achha.
Yeah. So that is the thing, right? The moment you put all these other particles, now I have to define the way the self-propelled particle interacts with them. Okay, so that itself is a challenging thing. These are not like collisional interactions like we do in granular matter or something, right? That is itself a problem. Now, if you say that I don't want to do that, I want to write an effective one-body problem, that is fine, but that effective one-body then has to be complicated. As I am seeing, one limit is active Brownian motion. The other limit is this noisy circle. In between, what is the effective dynamics? I don't even, cannot even describe by a single equation. Every density will have a separate equation actually. So it's a complicated thing if it is one body. But of course, simulations can be done provided we sit down and figure out what are the microscopic interactions we can.
Okay.
Yeah.
Okay, I'm sorry for the mic. The mic was.
No. I am also sorry that this whole thing. I'm still a little bit. We got delayed, and I was. Perhaps talking very fast. So do slow me down.
Okay, very nice. Nice. It's going on. Thanks.
Okay. So should I proceed to the next part, or any other question is there?
Yeah, I can.
So the circular motion happens because the concentration it diffuses much faster than other time scales. Right? So there's a time scale which is this characteristic time scale of the boat, and that's slower than the time scale of diffusion. That's why the boundaries become very important. Is it?
You could in a way say that. In fact, this analytical, nonlinear thing that is found, that actually shows that the circle that is produced, that is after stability analysis, that will be dependent on the size of the Petri dish. So it is not an independent thing. If the Petri dish actually becomes bigger, the circles also become bigger. Okay. So yes, the camphor diffuses off to the boundary, bounces back, and it's doing all kinds of things. Yes.
Yeah, because it was a little weird that the size of the shape of the boundary was actually so manifest in the actual trajectory.
Right. No, this boundary condition actually makes it means it is important. Yeah. So okay. So maybe I go ahead now. Is that fine?
Yes, sure, Dibyendu. You can go to the next.
Okay, so the second part, I will come to these sort of timekeeping problems in biology. So, there are these, the main problem I will be talking out throughout for the rest of the talk is the problem of lysis. So in this, this is the classic famous problem that viruses, they infect bacteria, and then after some time they grow in large numbers and burst the bacteria and go out. That is the lysis. Okay, but apart from that, there are other processes like bacterial cell division itself. So for example, E. coli, I mean, there is this protein FtsZ which accumulates, forms a ring and then contracts, and then there is the cell division. And many people now in recent works, they are telling that it's the number of FtsZ that crosses a threshold. So similarly, there are other problems I am also working on. At the end, I will mention these are like bacteria which enter vacuoles, and then they want to come out of the vacuoles. They produce toxin proteins which actually produces pores on the vacuoles, and those are also going to be regulated. After a certain time, the pores will be becoming big, and then the bacteria can come out. So all these types of processes, in a sense, I use the word as timekeeping processes. That after a certain desired time, some event happens in biology, and they happen when certain proteins sort of accumulate and cross a threshold. So for example, in this story of the lambda-phage virus infecting E. coli, the relevant protein will be Holin. Okay, so and in case of cell division of the E. coli, I was talking of FtsZ. In the other example, the toxin protein is the Pnemolysin for Streptococcus pneumonia, which is inside a vacuole. So, in all these cases, certain relevant protein is increasing and then crossing a threshold. Now, what is the similarity with what I was talking before? Now, this is also some kind of a random walk. You see why? Because the proteins, they will increase in number by some process. They will decay, just like a random walker can go forward and also come backward. And then the random walker reaches a boundary, and here the protein quantity reaches a threshold. So that is how you can imagine that in your mind you have to abstract that here is a random walk, not in real space, but a random walk in the space of protein number or concentrations. Okay, that's the analogy to keep in mind. And now, but that's already I talked in such a simple language, but biology is not that simple, and we should be reminded of complications or complexities that has been part and parcel of this field, and from which a simplified picture finally has been abstracted out. But still, it might be good to spend 5 minutes on telling about some sort of complexities that do exist for these biological systems. So first and foremost, this picture that the virus enters and the virus proliferates and they burst open the bacteria, this picture is true for virulent phages or virulent viruses. It is not true for temperate viruses. The one that I am talking about, lambda-phage, is much more complicated. What it does is all kinds of things. After entering, so this capsule shape is the bacteria. This virus, after it enters, it can actually make this bacteria go in 2 pathways. One is lytic or one is lysogenic. In case of lytic, that is what I already described. They increase in number, they burst open the bacteria. That's called cell lysis. On the other hand, the lysogenic pathway is one in which they enter and then they integrate their genome with the genome of the bacteria. So the bacteria becomes a new bacteria in our color, from the white capsule to the red capsule. So it's a new bacteria whose genome has changed because its genome has got integrated with the virus which entered, and that bacteria now divides. Okay, so this is the lysogenic pathway. How it will go to one or the other, there are lots of stochastic expression models, why this switch toggles and all that. It's none of our concern for this talk, but this is a very famous thing that happens for lambda-phage. Now the question is, why am I telling you things that I am at the same time telling not relevant for the talk? For one small reason, that is, this is how the experimentalists do the experiment. They actually take lysogenic bacteria inside which that viral genome is already embedded. They give a heat shock. Okay, they increase the temperature suddenly, and the bacteria suddenly becomes committed to the lytic pathway. But this way of doing, you see, what is the advantage? Why do the experimentalists do it? Because if they would have relied on the natural process, some of them will become lytic, then they don't know what is the time at which it starts becoming lytic, what is it doing. So the initial time is not under control. But if you are starting with lysogenic bacteria, giving a heat shock and producing the lysis, then you know what T is equal to 0 is, and that is a well-controlled process. So that is the
I missed one thing. So this process happens for all bacteria, or this is specific kind of bacteria?
This is a particular virus, lambda phage, which enters, when it enters E. coli, it does these funny things.
Just for E. coli, not for other ones.
No, lambda phage on other ones, I haven't even read literature. This lambda phage is a very special, I mean, on E. coli, most of the experiments are done. Yes.
Okay.
So for other viruses like T4, they wouldn't even do lysogenic cycle inside E. coli itself. Okay. So that is why lambda phage is special. And they use lambda phage because, this is like Drosophila, they have these, certain organisms they like a lot in biology, and they because for years they have been studying these.
But, I mean, but these two processes, these two ways of virus infecting bacteria, these two way, are there examples that other bacteria also, similar things also?
Temperate phage. Yeah. Other than lambda phage, there are other ones also. Yeah. That is there. But laboratory largely studied.
That is fine.
Yeah.
Just as information.
So there is but I am just trying to say is that by some means, if you want to forget this, forget it. But the point is that they actually start with a lysogenic bacteria, put a heat shock, then take it to the lytic cycle. That's all I am trying to say. So, but then what is this Holin protein doing, and what is its role? So what happens is the following. So this is the genome of the lambda phage. And there are, of course, on the genome, you find pieces of genes. So there is the Q gene, S gene, RZ gene. And this S gene, this code is shown here. Some of you who may recall that, the start codon AUG, from there, translation starts happening and you start producing, proteins. So here there are two AUG sites, M1 and M3. And so actually the same genome produces two proteins, S105, S107. One is called Holin, the other is called anti-Holin. So the anti-Holin actually tries to regulate that Holin does not do too much havoc. Sometimes anti-Holin suppresses its activity. So the biology actually very interestingly produces somebody who can actually suppress if Holin is, Holin turns out to be a corrosive protein, as we'll see. So this is a cartoon which will make it clear what is going on. So from the genome, these Holin proteins are being translated, and they come and deposit on the inner membrane of the bacteria. After some time, they actually come together and form huge clusters, and those clusters are kind of rafts that perforate the inner membrane. Okay, they puncture the membrane. Okay, how big are these punctures? About 100 nanometers. Okay. And through those punctures, another set of proteins called endolysin, which is produced from the R, they go out, they break the next layer, which is peptidoglycan. Okay, and then there are proteins that are produced by RZ and RZ1, which go and break the outer membrane. So there is a sequence of steps, but it turns out that the action of endolysin and the RZ proteins, those are very fast. So the rate limiting step is the corrosion done by Holin. That is why Holin is so famous in lambda phage story, that everybody says Holin goes to some level and then basically everything goes out of hand, it bursts. So, but you get the story that actually it is fairly complicated. When people talk of Holin, they are talking of Holin in excess of anti-Holin. So anti-Holin is also present, and sometimes modern experiments, I mean, very recent experiments, modern, I shouldn't say, everything is modern in the last 15 years, but what I'm trying to say is very recent experiments have even tried to get rid of, they produce mutants where there are no anti-Holins, so that they can see purely the effect of Holins. That also is there. So cleaner experiments can be done. But then once these Holins are there, question is that what are they, if it is a biophysical process, then I have to know the geographically what these Holins are doing. Are they forming small clusters? Then they are forming large clusters. Then they are going and sitting on the membrane. What are they doing? Those can be very complicated things. People speculated, people did experiments, 10 years passed. Then in 2011, a very decisive experiment came, which kind of made it very crystal clear what is going on. What I can't show all the pictures of this very fantastic paper which appeared in PNAS 2011. What they actually showed was that Holin comes and uniformly deposits on the surface for a long time. Then within a few minutes, and here minutes are shown by 93, 94, 95, you can see nothing was there in 93. In 94, suddenly a green blob. This green blob is that all the proteins on the membrane, they are rushing to one spot and they are making one huge cluster. And by 96, and after that, boom, the whole thing bursts. Okay, so this paper actually basically showed that all those people, they were breaking their head about the biophysics, can go out of business. The only thing that is happening here is that some number is crossing a critical number, and once that happens, then the thing goes lighting. So the excess number of proteins, these were also estimated. About 1500 excess Holins have to be there, and the time scales that they quote are total lysis time is 65 minutes, out of which 15 minutes have to be taken out because initial 15 minutes is what is called a late promoter activity, during which there is no production of Holin. Holin starts being produced after 15 minutes of induction. So the 65 minutes is measured from the time point of induction. But after 15 minutes, Holin production starts. And then in 50 minutes, roughly this process happens.
Just to be clear, so antiholin is not produced in this experiment, right?
Yeah. Antiholin is being produced. So this particular 1995 paper that I am talking about, this is much earlier to this 2011 paper. They estimated antiholin and holin together, that was about 4,500. So I told you 2:1 ratio, right? So this is like 1500 excess Holin. When I say Holin, I mean excess Holin. I'm forgetting antiholin. Yeah. So now that being the case, now when a biologist tells you, oh, we know lysis is happening in wild type lambda phage infecting E. coli 50 minutes, don't trust them. Okay, when they say 50 minutes, they may mean that, well, sometimes I do experiment, it is 60. Sometimes I do experiment, it is 40. Okay, on an average 50, I am happy with 50. But that means that if you want quantitative biology, you would be interested in the fluctuations of this time. Okay, naturally. So then one has to get into mathematical formulation of the problem. One has to first of all know how the proteins grow and shrink, decay in time. You also have to know that, once the proteins cross a threshold, we are saying first passage or lysis is happening, that time is important for us. What are the fluctuations of these times? And if possible, what is the distribution of those time scales? So to mathematically formulate it, one has to know what is the process by which protein is produced, and that. So this quantitatively was done in, I was very amazed, this group of Harvard, they wrote 3 papers in the same year, 2006. One Nature, one Science, one PRL. And in the Nature and Science, they showed that generically in E. coli, the proteins are being produced in the translation process in bursts. Okay. So it is not that protein number goes from n to n plus 1, but n to n plus r. And this change of r, the likelihood of going to n plus r from n is exponential. Okay, so it is exponentially, so as the r will be larger, it will become more and more unlikely to go there. But there is a certain burst size, mean burst size b, and this is a distribution. So once this is done, so at least experiment is telling you what is the mathematical form of production that you have to put. Also, with this form, these people wrote down the continuum master equation and actually proved that in the steady state, the protein distribution, the protein concentration distribution is going to be a gamma distribution. Some of you might have heard this very famous thing that protein distributions often are gamma distributions. So this is where this thing came from. Single year, single group, writing all these things together. Okay, and 2 years later, the discrete version was also written out very nicely. And so what is it that it is telling? So let me explain the terms. This is a forward master equation. Pn denotes the probability that you have n proteins at a particular instant of time. So you can come to the n protein from any protein that is below, starting from 0. So you see, for that to come, the rate, of course, will be exponentially decaying the further and further you go. So that is the b divided by b plus 1 to the power r factor. So this is a discrete geometric distribution corresponding to the exponential distribution. And there is a transcription rate, which is k. You can say why the transcription rate has a subscript. Isn't it constant? Well, it turns out that people kept the possibility that there can be feedbacks. So if there are feedbacks, the protein number state actually can control whether the transcription rate will be small or large, depending upon whether the feedback is positive or negative. So this is the most general model that one wrote down. So transcription rate multiplied by the burst or protein burst probability, that's the net rate of production of protein. And that is multiplied by the probability at n minus r. That's the term which is causing the inflow to the state n. On the other hand, another inflow is there from a higher state n plus 1. So if you have n plus 1 protein, one of the protein can decay, and that's how you can get n. So that is the third term. Okay, so the first term and the third term are the inflows. The second and the fourth are the outflow terms that basically denotes that from the state n it can go to n minus 1. That's the fourth term. And the third term is that from n it can go out to all higher states higher than n. Okay, that's the second term. So this is simply the master equation. And that being the case, what is known in this literature? So this is the very gross picture I will first show you. That is, the protein number is increasing as a function of time. And then you can ask the conventional question that in gene expression people ask. That is that at a given time t prime, what is the distribution of the number of proteins? That is written on the left side, p n t prime. And that is the conventional question, right? Time is fixed, you ask for the number. On the other hand, in recent years, this other question is also being asked, which is that somebody fixes a threshold of the protein number, that is capital X, and you ask, what is the time for the first time, what is the distribution of the times at which for the first time that threshold is reached? Okay, so the X is attained by the NT function at certain time t, right? But then the t is not the same. Different times it reaches at, or different samples reaches at different times. So you would be interested in the distribution of the times. So that is the first passage time distribution. So we found out that in 2017, this problem for the first time, its mean and variance was worked out in this PNAS paper. And Abhuday Singh's group in Delaware, they had worked out this mean and variance. So, but then the distribution, it turns out that nobody had done, and this is in 2020 and 2022, we have solved this problem for the full distribution. So the star student, Krishna, who is there in the audience, he has done all this beautiful work. You know, I must laud, because he's one of the very bright students I have got in recent times. And this is my old friend Ashok, and he is also older than me, as you can see from his white hair. And you know, the way to handle this, what we realize first is that first passage can be obtained as a time derivative of survival probability. So what is survival probability? Survival probability is that you can start from some state. Here, suppose the subscript is S0. You start from 0 number of proteins, and your protein number is increasing, but you survive not reaching the threshold up to time t. That is survival probability. And first passage is that between t to t plus dt, you have reached it for the first time. So, you know, one is the derivative of the other. So if one can know survival probability, then one can know first passage. So what is the equation for survival probability? So survival probability equation very easily can be written in terms of what is called the backward master equation. So what is a backward master equation? So as some of you may know, usual forward master equation is that I know where I am at the present, then I ask where I can go in the future. So that is forward in time. But this equation is backward. That means t plus delta t here is actually the past. T here is future. Okay. So basically you will see, if you compare this equation with the previous equation I had written, you see in the previous equation I was telling you from which states I can come into N. But because I am going in the past, essentially that will get inverted. I have to tell you from states N, where do I go at the next time instant. That is from at t plus delta t, I have to go to t. That is from past to future. Then I go out from N to different states. So that's all. So basically the signs get reversed and all, and this is a standard procedure, but just mentioning that this is a backward formalism. So this kind of equation can be casted in a matrix vector equation, and then this is a first order equation which you can take a Laplace transform of, and once you take Laplace transform it becomes an algebraic equation. So a particular matrix has to be inverted, then we know the survival probability. What kind of matrix? The matrix looks a bit horrible like this. I stared at it for some time, but then you have a genius like Krishna who can see patterns, and then basically he basically found out that for xth order what will be the inverses he guessed, and then he told me that this is my guess. I sat down and proved that these are proper inverses. Various rigorous things we proved, but the first guesses all came from Krishna. Okay, so but anyway, the point is that you can solve the problem in Laplace space, and then you can invert back in time, and then you can get the full answer. So what could we do? Could we solve everything? Well, it turns out when degradation is 0, we could get not only the Laplace transform, but the time domain answer also. That is, we could invert the Laplace transforms. And these are basically some complicated expressions, but essentially a bunch of some exponentials and sometimes multiplied by some power laws. But then that would mean that the tails of the distributions are non-Gaussian. They are exponential tailed. And here was that published data for the Lysis times of the wild type lambda fudge. Blue is the graph that we have from theory. And we just plotted it because all the numbers, otherwise capital X, everything, this came from experiments. We just had to put those numbers in our theoretical formula and plot it. And this is the graph. Initially, we were happy. Everything is nicely, it seems falling on top of each other. Only we were little unhappy immediately seeing that if we plotted just a Gaussian, that is the red dashed curve. So the Gaussian, forgetting all these complicated calculations, if you just take the mean and variance and plot a Gaussian, that also fits the data quite well. So what is the big deal? What is the big deal of calculating so big a calculation? I mean, so big a formula. So, well, I mean, then we started thinking that are there things that can be predicted which will be non-trivial from our theory. So one of the thing we sort of said was that CV square should monotonically decay with mean first passage time. So how to test that? No way. You have only one point. Experimentalist has given us the wild type. Okay. So that is one point. With 1 point, how can I test a graph? So we said, okay, fine. What can we predict from, degradation rate being not equal to 0? Well, for that, we only managed to get the Laplace transform. We could not invert it in time, but you can numerically invert it. But forget numerical inversion, you can use it to get the second order fluctuation, which is CV square. CV square, I remind you, is variance divided by mean square. So that is plotted against mean first passage time. And that shows a non-monotonic behavior. We were excited. Non-monotonic behavior is something we have seen elsewhere also. But again, you see, the problem is that we have only one data point. How can we check a curve with one data point? And why this non-monotonic behavior is happening here, let me tell you, is that mean first passage time will rise if the threshold rises. Because, if the target is further away, it will take longer mean time. But the system, given some production rate and decay rate, will have a steady state. So because it will have a steady state, it will have a average protein number in the steady state. If your threshold is going beyond that, the system will find it very hard to attain those targets. And as a result, the time scales will be coming from rare events and hence fluctuations will rise. So that's the cause of this non-monotonicity. It has got nothing to do with the non-monotonicity that we saw in the Camphor Boat, but it has its own reason for this non-monotonic fluctuation. But you can stare at such graphs, you can have wishful thinking, if somebody could give me some more data points, I could fit it. But then we saw that all that was done in this paper. So this is the dream curve that came in 2020, again from Abhyuday Singh and his collaborators. This was published in iScience. What you see is that these guys actually took 20 genetic mutants of lambda fudge. Each one of them have different mean times. Mean times because they have mean thresholds. They have thresholds of proteins after which lysis happens different in the different mutants. And as a result, since the threshold varies, the mean also varies. But interestingly, this non-monotonic fluctuation. But that immediately puzzled us, and we contacted these guys that what is going on. It is impossible that you can get non-monotonic behavior because you remember we showed it here. If Holin is what you are using, Holin does not decay, its decay constant is 0, then the curve should be monotonic. How come you are getting a curve which will correspond to non-zero decay constant? They said, because we have now understood that protein number is not the target, protein concentration is the target. Now that was a completely new feature that they had. What do they mean? I will come in the next slide to make it clear to you. But bear with me that there is some completely new understanding now that these things are crossing a threshold, not of a number, but of a concentration. Okay. And moreover, we saw that what they had done in the paper, they forgot all the burst. They just wrote down some approximate Langevin equation in which there is a constant birth and some decay. And they sort of even did not solve it exactly, and that was the state of the paper so far as theory was concerned. When we contacted them, they said, okay, we have nothing. We don't have even mean, we don't have any variance, nothing exact. Okay, forget for distribution, we don't even have any moments exactly calculated. And we were very distinct that this seems to be something simple, why haven't they calculated? But anyway, then he told me why he did not calculate, because, and this is the new work in 2020 which has appeared in Physical Review Letters in collaboration now with Abhyuday Singh, who gave us his published data, and that's why we could compare. None of the new experiments were done. These were all published data, but he gave us kindly all those and he became our collaborator. So what he reminded was that, look, Holin is not decaying, but look at the concentration. Concentration is number by volume. The cell volume is increasing. Cell volume is increasing, and as a result, what is happening is that the concentration that is going to decay. Okay, so at what rate? That also will come from the cell doubling time. So if in 40 minutes the cell doubles in size, then this would be the decay constant that you should use in your theory. So that was a very fantastic insight that they gave, by doing which, by approximately plotting their theoretical curve, they were getting this experimental non-monotonic behavior. So what he told me is that, can you solve the continuum problem? You guys have solved the discrete problem in the PRE. Can you please solve this continuum problem? And then he said the continuous equation in the forward master equation was already written in the Physical Review Letter 2006. So can you do your backward calculation? And that is what we did. We wrote the backward equation corresponding to the forward one for the survival probability. It is an integral differential equation, unlike the other discrete equation where matrices were involved. Now we are dealing with function spaces and of course, continuous variables and everything. And now, but it is a master equation. The first term is a jump term with exponentially jump things, and the second term are continuous, so basically it is not a Fokker-Planck equation, not a full master equation. It is what you may call as Chapman-Kolmogorov equation and a backward one. Okay, so with that, we started, and then, you have to take derivatives of these, and then you will get a partial differential equation. And then in the time domain, you have to go to the Laplace space, and then you get an ordinary differential equation in concentration, which I have written here, very well known from our MSc physics. This is the confluent hypergeometric equation. You just then find the exact solution in one step. Okay, so this is the full formula in Laplace space for the first passage time distribution. Now the question is to match with experimental data. We had to fix the parameters. So what were the parameters we should look at? Decay constant, as I said, is purely coming from cell doubling rate. The initial concentration of the proteins are near zero. So two of the parameters are fixed. Then remains the transcription rate and the threshold. Every mutant has its own threshold. So depending upon the mean, if the mean is large, threshold is large. If the mean is small, threshold is small, but it varies. But what does not vary is the transcription rate. How do you know that? So in this iScience experiment, they actually presented, Western blot. So these are basically concentration of proteins you can get from the wholesale extract as well as from the membranes. So what they saw is that inside in the bulk, all the mutants were having similar kind of protein concentrations, but the protein concentration was varying in the membrane enormously. So the mutants were different mutants simply because the attachment and effectivity in the membrane were different. The production rate of the proteins were not different. So the K constant can be treated as a constant through all the mutants. That was great because then you have a single fitting parameter for 20 data sets that I have. Each one of these data sets, are coming from 150 experiments and all that. So basically a single data fit, that I stress, that is what makes us very confident. And this is what it is. There are 20 mutants. These are the histograms. The black curves are the theory curves, and the orange things are the histograms from experiments. And what we liked very much was that if you looked at mutants on the extreme right column, you see these curves are skewed and they have long tails. See, this is what we were looking for, that in some data, if there is huge deviation from the Gaussian, then only it makes sense to do all these hard calculations. Otherwise, people will say, I know mean, I know variance, why do I care for the distribution? But you need to care for distribution if you have skewed graphs like this. And then we actually fitted the skewness now with the data. So this is the data and this is our theoretical graph. As you can see, variance was already fitted before. And then we also went back and asked that these guys in iScience, they had done an approximate theory. Can you derive the approximate theory from the exact theory? So what we did is what is called a systematic Kramers-Moyal expansion, in which you can go from a master equation to a Fokker-Planck equation, and that is up to keeping the second order fluctuations. And that is what we did. And we got the Fokker-Planck and the corresponding Langevin they had used in their paper. Not only that, that Fokker-Planck I exactly solved. This is the font. And this is what is the comparison between exact theory and approximate theory. If you go to second order, then exact theory and approximate theory are same. That is the black graph and the red graph are the same. If you go to third order, that is skewness, you will see exact theory is starting to deviate from the approximate theory, which is expected. And the simulations are Langevin simulations, but Langevin simulation is corresponding to the Fokker-Planck equation, which is itself approximate. So no wonder the simulation will match the approximate theory. If you do a simulation, which is like kinetic Monte Carlo, which will simulate the master equation, that will, of course, be corresponding to the exact theory. Okay, so this is the conclusion. I think I have finished in time, if you have pardoned me my 15 minutes overshoot. So the conclusion is that we obtained exact distribution of threshold crossing times of protein numbers as well as concentrations, and applied them to the problems of lysis times of bacteria after lambda-phage infection due to Holin accumulation. Okay, this was a concrete case where we had data where we applied, and not the non-monotonic fluctuations in the lysis time with varying concentration threshold. That non-monotonic behavior we could actually explain from theory, and not just to second order. This non-monotonic behavior not only in skewness, we have even looked at, I am not showing you data, that the exponential tails, the characteristic time associated with the exponential tails, those also show non-monotonic behavior, and we have extracted those from experimental data and plotted with theory, they also match. So, this is fantastic, because all the large deviations and everything we can match now, because those were not possible by just merely first two moment calculations. So with Anirban Banerjee of the Bioscience Department at IIT Bombay, I am working on this problem of Streptococcus pneumonia, which has to cross the epithelial layer, which is often called the blood-brain barrier, because on one side there is blood, on the other side is the brain, and if this pneumonia bacteria, they cross the epithelial layer and go to the brain side, you have meningitis. So, but how do they enter the epithelial layer? They put themselves inside a bag, which is called a vacuole. And they stay inside the vacuole, but it is its nature to produce some toxin proteins called Pnemolysin. Some of the subpopulation of the bacteria produce in large amount this Pnemolysin. Some of them produce in small amounts. And this is a puzzle in biology. Why do they have subpopulations? What is their population survival strategy and all that? That's another problem we are working on. But at least at the level that we have data about when do the pores form, the Pnemolysin corrodes the vacuole wall, and when the pores form, that kind of time scale data we have from experiments. So we are trying to fit our kind of discrete theory that we have to those data. So that is some ongoing work, and if in future I again visit, I will tell you about that. And thank you for your attention.
Yeah, okay. Very nice talk, Dibyendu. Very nice.
Thank you.
Yeah. So in the Langevin equation there is this KB. Just before that, there is this KB. What was that KB? dC by dt equal to.
So this is this B. You remember? This is the mean burst size.
Achha.
K is that transcription rate. So instead of, see, the original actual problem does not have a constant rate of production, means like it is not just constant, right? It depends on the jump state. All that is gone in the smoothened version. Master equation has a jump process. This has become like a smooth process with just a burst number. And then the decay term is gamma times C, which is expected. That is fine. But the whole jumpiness that was there in the master equation, that has gone away in this smooth description of the Fokker-Planck and Langevin.
But still it is working. That is very
well. I mean, no, it is working to that level that they looked at only second order fluctuations. It will not work, because if you go to third order, it will not work. Because, then the exact theory is needed. That was our whole struggle through the paper. That everybody tells that we know mean and variance, and that's in the end of the story. And we were struggling to prove that there are instances where you require to know higher order fluctuations. Yeah. So that's the.
And this other page, second page, can you go to the next page?
One thing, Dibyendu, what are these are two different. So this is the same data. In one case, you are plotting the third order skewness, right?
Yes, the third order fluctuation. See, the third order fluctuation cannot be calculated from this Fokker-Planck theory. For that, you have to go to our exact theory, is what we are saying, right?
Okay.
Because the third order fluctuation, if you force to calculate, then the lower graph is showing you will get wrong answer. So that is what we are saying, that you can, you have a theory which is approximate. You can force it to produce answers which it is not supposed to produce. You will get wrong answers, of course. But if you do the right theory, you will get a right answer. Then you can match it to experiment. Is the point.
Next page, please. Next one. The page where you match this. No, not the one where a lot of data has been matched.
Yeah, this is really very impressive.
Well, I mean, we were very impressed because there is a single fitting parameter. I am not really fudging anything here. We were amazed when they gave the data. This guy told that we can't do. And then, this is so hard to do the work sitting here. We had certain other predictions also. We told that if the experimentalists can be brought in, we can test those. So then they say, no, if we bring them in, it will become another story. Maybe we'll try for the next paper. So then you live with these dissatisfactions that you have predictions that can be tested. But this was very impressive that there is a single parameter, actually.
Yeah, that is the reason.
Actually, some of these, the experimental histograms, I see have both the left skewness as well as right skewness. This is very weird, no? Like,
no, this graph that we are having, it will have a skewness on either side, because the distribution is funny.
Oh, I see.
So,
okay. This is not like a gamma kind of a function that have only.
No, gamma will also have. It will have an exponential tail on one side and a power law build up on the other side. So you will have asymmetry on the two sides. ऐसा तो नहीं है कि you will not have. Yeah. So, but PK was telling something.
No. That is all. That is very, very
okay.
nice.
Yeah. Okay. Yeah.
Anyone else?
Okay, so if there are no other questions, I think we should thank Dibyendu.
Okay, I'll just maybe open the this thing, stop presenting, and then so I can just to everybody. It was very nice that I could come and chat with